And our next speaker is Dena Leerberg, who's going to tell us about FGF24 and gonad development.
Good morning, and thank you for being here. We in the Draper Lab are particularly interested in gonad development simply because it is the most important organ in the body. Evolution agrees with us. So zebrafish gonad development occurs in a stepwise fashion that is very similar to that of other vertebrates. We begin with the specification of somatic gonad precursors that give rise to the cells that regulate germ cell development, and primordial germ cells, the gamete lineage. In zebrafish, PGCs are specified during the mid-blastula stage, as we just heard about, and they begin to interact with somatic gonad precursors at 5 days post-fertilization, or DPF, and this is a phase that we've called, that is termed initiation. This gonadal primordium will then undergo a period of growth, and this phase lasts a couple of weeks in the zebrafish. At this point, the gonad is bipotential and will only differentiate into an ovary or testis during the process of sex differentiation. And in the zebrafish, we find that we can start to see sex-specific differences in the gonad by about 20 days post-fertilization. Well, we know a lot about this last process based on work in the zebrafish and other vertebrates. The processes, the genetic factors that regulate these early stages of initiation and growth is fairly lacking, with the exception of a few transcription factors that are known to regulate mouse gonad development. And so these are the phases that I'm really interested in learning about. Specifically today, I'll tell you about a novel role that we have discovered for FGF signaling during the growth phase. Specifically, we have found that a mutation in the fibroblast growth factor 24 ligand results in underdeveloped larval gonads, depicted here. In these top images, I'm showing confocal images of isolated gonads stained with the germ cell-specific VASA and DAPI. And we can see that the homozygous mutants have a smaller gonad in comparison to wild type. We can further count the number of germ cells per animal and see that while wild types and mutants begin with the same number of germ cells, while wild type animals increase their number of germ cells over the next few days, mutants increase their germ cells only minimally. This is even more obvious by 40 days post-fertilization, where mutants have only scattered germ cells across their pitiful gonad, and this is in comparison to the wild-type testis that is just chock full of germ cells. At a higher mag, we can see that the germ cells expressing GFP here are surrounded by somatic cells. These somatic cells are expressing the transgene GSDF-M cherry, which is known to label both Sertoli cells in males and granulosa cells in females. In contrast, in the mutant, we see that the somatic cells that associate with the germ cells do not express this GSDF, suggesting that these somatic gonad cells that we see in the wild type are either absent or dysfunctional in FGF24 mutants. This led us to the hypothesis that FGF24 may in fact be required for somatic gonad development, and that the germ cell defect that we see is really secondary. We reasoned that if this was the case, we might begin to see somatic gonad differences, defects, before we see a germ cell defect. We therefore performed in situ hybridization to look at the transcription, look at the expression of GATA4, a transcription factor known to be required for mouse gonad development, and CYP19A1A and AMH, which are female and male somatic gonad markers, respectively. I have outlined the gonad based on VASA expression, and we can see that these three genes in purple here are expressed in most of the wild-type gonads, but rarely in the mutant gonads. Because these time points precede the germ cell defect that we've seen, these data support our hypothesis that FGF24 is actually required for somatic gonad development. We were next interested in which cells are expressing FGF24, and fluorescent in situ hybridization shows us at 10 days post-fertilization that FGF24 in orange is expressed in somatic cells and not in these magenta labeled germ cells. We further wanted to identify which population of cells was responding directly to FGF24, so we looked at the expression of ETV4, or also known as P3, as this is a gene whose transcription is known to be downstream of FGF signaling in several contexts. In green, we see that ETV4 is expressed in a distinct layer of somatic gonad cells, and this expression is dependent on FGF24 signaling. So this leads us to this model that we're forming where FGF24, we can see in this orthogonal view of this gonad, that FGF24 is expressed in an outer layer of somatic gonad cells, and this is signaling to an inner layer of ETV4 expressing cells, which then are in contact with the germ cells. This was a new structure to us, so we wanted to look at it a little closer by EM. And in this transverse section of a 10-day-old wild-type gonad, I've pseudo-colored a single germ cell magenta in the center. This germ cell is surrounded by a thick layer of somatic tissue, and in agreement with the fluorescent in situ data that I just showed you, the somatic layer seems to be separated into two distinct layers, where the outer layer is likely the FGF24 expressing layer, and this inner layer would be the ETV4 expressing layer. Furthermore, if we take a closer look, these layers are separated by this electron-lucent space, which made us wonder whether there was a basement membrane forming. Indeed, immunostaining showed us that laminin, one of the central components of the basal lamina, and shown here in green, is localized to the circumference of the gonad, and often we see it between these somatic gonad cells, supporting our hypothesis of a forming basement membrane. In contrast, in the FGF24 mutant gonad, while we still see germ cells enveloped by somatic gonad cells, the somatic portion is no longer split into this bilayer, and we never see this electron-lucent gap. And accordingly, we have not been able to detect laminin deposition in any of these gonads. So to sum up so far, we have discovered that the 10-day-old wild-type gonad is actually already multi-layered, where an outer layer expresses FGF24 and seems to induce transcriptional changes in an inner layer of somatic gonad cells, which would be like the ETV4 expressing layer. And this signaling is required to promote morphogenesis of the early gonad, as well as to promote germ cell development, as both of these processes are defective in the mutants. So I would next like to tell you about our endeavors in trying to identify which FGF receptor is responsible for transducing the FGF24 signal. I'll remind you that FGFs are secreted ligands, generally secreted ligands that bind transmembrane FGF receptors to bring about cellular responses. And if we think about this cell here, we would hypothesize that in our case, this would be the inner layer of somatic gonad cells. The zebrafish genome contains 5 FGF receptors, FGFR1A, 1B, 2, 3, and 4. And to somewhat of a surprise, we found that all 5 receptors are readily detectable by RT-PCR in the 15-day-old wild-type gonad. So our approach then was to use CRISPR-Cas to generate null mutations in each one of these FGF receptors singly and look for a gonad phenotype. However, to our surprise, all of the receptor mutants were embryonic viable and had no gonad defect. So after a lot of head shaking, we concluded that multiple FGF receptors must be able to bind FGF24 and are working in a redundant or compensatory manner. So we are currently in the process of making all of the combinations of these mutant receptors to see if we can find a phenotype. And so far, we have data concerning the FGFR3, FGFR4 double mutants, which I will share with you now. In situ hybridization shows us that at both FGFR3 and FGFR4, shown here in green, are expressed in somatic gonad cells that abut the germ cells. And I'll remind you that this is the population of cells that we're interested in this experiment. So when we're looking at a phenotype that phenocopies the FGF24 mutant, I'll remind you that we're looking for a smaller larval gonad in comparison to wild type, and one that has significantly fewer germ cells than wild type. So when we mutated both FGFR3 and FGFR4 in animals that were homozygous wild type for FGF24, we actually did not see a significant decrease in germ cell number or gonad size compared to wild type. However, when we mutated both receptors in the FGF24 heterozygous background, we now saw a decreased gonad size and significantly fewer germ cells compared to wild type, and very similar to that of the FGF24 mutant gonad. So while it's clear that these are not the only receptors at play, it does appear that they're playing a role in this pathway. So overall, I've told you that FGF24 is expressed in an outer layer of somatic gonad cells in the 10-day-old gonad, and signals to an inner layer to induce transcriptional changes of several gonad genes. This pathway seems to be at least mediated by FGFR3 and FGFR4, although these are certainly not the only receptors at play. And this pathway is required to promote morphogenesis of the early gonad as well as promote germ cell development. And so overall, we have shown this is the first report of FGF signaling being involved in these early stages of vertebrate gonad development. However, I will say that when the human orthologs of ETV4 and all of the FGF receptors are amplified, that is correlated with aggressive ovarian cancer. And so something that we're starting to consider is that the pathways we've uncovered here in early development may be co-opted later on to promote tumorigenesis. With that, I'd like to thank my wonderful lab, particularly my excellent PI, Bruce. He is giving a sex determination talk on Saturday that I encourage you to go to. And I'll point out also that Yana and Michelle are giving posters this week on different aspects of gametogenesis and reproduction. And with that, I'll take questions. Thank you.
Hi. Did you look at these ETV and FGF24 cells later in the ovaries, or?
Yes, so later on, it appears that ETV4 may actually be specifying a sort of a precursor somatic cell in like the early juvenile stage. And FGF24 remains expressed in an outer layer that it may be expressed in like the epithelial cells.
So you can still detect two distinct cell populations.
Mm-hmm. Yes.
And so do you think that the FGF24 ones somehow act in an endocrine manner to become the ETV4, or these are two separate?
At least at this point, it's unclear later on how they're interacting, but at least at this point it seems like it's acting in a paracrine manner to signal to the ETV4 expressing cells and initiate transcriptional responses.
Could you associate them with germline stem cells later?
No, so while they may be regulating the germline stem cells later on, it doesn't appear that they are germ cell expressed, that these genes are expressed in the germline stem cells.
Right, they could provide a great niche for
yes,
them.
yeah, that is possible, but something that we certainly haven't shown.
Next.
Hi, quick question about your basement membrane data. It was a little unclear to me which specific cells are in contact with the laminin layer that you showed, and I'm curious to know, without that laminin there, are those cells still present, or are they lost in the absence of that ECM?
In the mutants, did you mean the mutants?
Yeah, I'm sorry, in the FGF24 mutants.
Yeah,
Yes.
so it's actually unclear. What I didn't point out is that we don't have really sufficient markers at this point to determine whether the remaining somatic cells are the cells that are more like the FGF24 expressing layer, or whether it's a mixture of the populations, or if it's some sort of precursor cell type. Yeah, so it's still really unclear.